All right. It is 2. We're recording, so I'll get started. All right. And so reminder, next week on Monday, the 12th, Big Bad Test 3. From this week on, the material we're going to cover won't be on test 3, but will be on test 4. So all the material that for test 3 is already out there in the folders. Study guides are out there as well. And I need to put up all the answers for those. I don't think I've done that yet. As a reminder, how best to study for the test, what I would do is I would first make sure I have the study guides in front of me, but with no answers in them. Go through the study guides, see if you can answer everything without looking back at your notes. If you can't, then that's what will tell you what you need to focus on. So you go back and look at that stuff, see if you can answer it again. Then go through the notes again, look at the problems I ask on there. Can you do those? And if you can answer all that, and then if you're still looking for stuff to do, go back to the homework, see if you can answer that. Go through the slides one more time, see if you're good with that. You should have no problem on the test. That being said, the best way to go about it is to start right now. Well, it would have been great to start last month. You keep doing the study guides on and on and on. But if you haven't delved into it yet, don't wait until Sunday. Start right now and do a little bit at a time. So like normal, the test will be open for 24 hours on Monday. You have 1 hour to take it. And we're going to use monitoring software again, just to try to keep the integrity of the class. Yeah, that's test 3, and that is next week, Monday. But let's get into the material that's on test 4 then. And we're going to continue to talk about the membrane, the lipid mosaic membrane. And this slide is combining a lot of the stuff that I went over in the videos. So let's just go over some of the stuff we're looking at. First, on the bottom left here is just our mosaic model of the membrane. So it's a lipid mosaic model. What does that mean? Well, that means that the membrane is not just full of lipids and nothing else. It has some compartmentalization with it. Different parts of the membrane have different features, and different proteins are associated with different parts of the membrane. Here we can just see how varied our membranes can be. So here we have some glycolipids. Remember, glyco means sugar. Whoops. So that is just showing that some of our lipids have sugar on them, and these are usually on the extracellular portion. We use these for signaling. Here, peripheral proteins. These are proteins that only bind sometimes bound, sometimes not bound. So they come on and off. Phospholipids. Remember, we talked about how there's a lot of different lipids. We talked about sphingolipids, cerebrosides. There's a lot of different lipids out there. So they're just not all one type of lipid either. Got your cholesterol in the yellow here as well. Remember, that's to help your membrane become either more or less fluid-like. Here's one of our lipid-like proteins, where we have a lipid attached to our protein, which we talked about last week. The lipid is embedded in the membrane. Now this protein is also embedded in the membrane as well. Here we have what's called an integral protein. Integral means it can't leave. It can only function in the protein. And this is also a transmembrane protein. And that it goes all the way through the membrane. Here we have an alpha helix, hydrophobic. That's how a lot of integral membrane proteins are. They have an alpha helix. Remember, all this is nonpolar. So all the amino acids here will be nonpolar. However, something that I didn't mention before, and I really should have, the reason why it's like this, like an alpha helix and not just a random string of amino acids, is I remember that all amino acids have charged termini. So here's alanine. So it doesn't matter if you're hydrophobic or not for the side chain. You always have these charged termini. And those charged termini would not be happy in the lipid membrane. Even in a peptide bond, where you're no longer charged, I guess that's really how I should talk about it. You can see we still have polar groups. And these polar groups really don't want to be in the membrane either. So by having this alpha helix, your backbone is hydrogen bonding with itself. Remember, we talked about secondary structure of alpha helixes, how there's a N+4 relationship for hydrogen bonding. So residue 1, hydrogen bonds with residue 5, that's the N+4, right? And so that's how we can have something polar, like a protein backbone, be in the membrane by that alpha helix. You can also have beta barrels. We showed the picture of that where you have like beta sheets that go through the membrane. So be like almost something like that, except these would be like beta sheets, and then there's like a hole in the middle of this where you can let other things pass through. So that's another type of integral membrane. But yeah, it's very varied, right? That's our lipid mosaic or fluid mosaic model. Right. So that's what we're going to be talking about today. Now let's look at some experiments that was used to determine the lipid fluid mosaic model of the lipid bilayer membrane. This first experiment is called FRAP, which is fluorescence recovery after photobleaching. Another way to call this is Forster or something like that, recovery after photobleaching. Might have the name wrong, but people call the F different things. I use fluorescence because it's easy to remember. All right, so first we have to talk about how this experiment works before we can actually talk about like what it's doing. So for things that fluoresce, let's take GFP for example. GFP is a protein that fluorescence that glows green when you shoot it with a certain wavelength of light. That's called fluorescence, right? Light comes in, UV or whatever, light comes out, right? That's fluorescence. Now, one way to kill fluorescence is that if you shoot a laser at a fluorophore is the thing that fluoresces. And GFP, I think we might have looked at the structure of that in one of the videos and not talked about it in class. But it's basically made out of 3 amino acids that form a new structure that allows GFP to be fluorescent. Well, what you can do is that you can kill fluorescence by hitting fluorophore with a laser, right? So you no longer get this. Okay. Now let's see how we can use that. So imagine that we're looking at a cell. And let's say we took every single lipid in the cell and we made it fluorescent. So every lipid in the membrane rather made it fluorescent. And then we look at the fluorescence and we see, oh, okay, it's green all over the place. Good, we have lipids everywhere. then we hit a part of the membrane with the laser, killing fluorescence. So we get this giant white spot. And then we wait some time, which is called recovery. And what we'll see is that this white stuff, which are the lipids with dead fluorophores, they will move out of the circle just due to random diffusion. And the lipids with fluorophores will move into this dead spot, which means lipids are moving. Which means that a lipid in the membrane isn't just stuck in one place. You can also do this with proteins, and that's a common thing for proteins, where you, if you want to measure something like diffusion speed, that's another good use for FRAP, is that if you're looking at a specific protein in the cell, you make it so all the copies have a fluorophore on it. Then at a certain part of the cell, you kill those fluorophores with a laser, and you just measure how long does it take for proteins to go back into that circle. That's measuring diffusion speed. That's measuring how fast proteins move inside of a cell. And it's the same technique, FRAP. But yeah, that's the basic technique of FRAP there. And here's a graph that's showing the same thing as this picture, where we have fluorescence, fluorescence, fluorescence. And if we're just looking at a certain part of the cell, we bleach it. So that part of the cell no longer has fluorescence. And as long as whatever molecule has fluorescence that you're looking at, that fluorescence signal will slowly recover because it's free to diffuse. If you had a protein or a molecule that wasn't free to diffuse, right? Your line would just look like that because nothing could move into that spot. So that's FRAP. That's one way you can tell that the lipid was fluid. Another way is shown in this experiment. So in this experiment, we have two cells, mouse cells and human cells. You color the mouse cells with a green fluorophore, and the proteins on the mouse cell with the green fluorophore. So this is proteins. And for the red cell, all the proteins are colored, or on the human cell, all the proteins are colored red, right? So that's the start of the experiment. Two separate cells, one's a mouse, one's a human. You just color each protein on the surface of the cell. Then you mix the cells together, and with help of a virus, you can make these proteins fuse. And what you see, as shown in this picture, immediately after fusion, the proteins are cell segregated, right? All the human proteins are on one side of the cell, all the mouse proteins on the other side of the cell. However, give them some time, so 40 minutes at 37 degrees C, and you'll see that all the proteins have dispersed. All right, there's now an even mixture of red and green proteins, which tells us proteins are free to move in the membrane. That's a big takeaway here. And I should say most proteins, not all. Most proteins are free to move in the membrane. Again, showing us that our lipid membrane is just not static. There's movement to it, and it's varied. You have lipids, you have proteins, you have different types of lipids, you have different types of proteins. And that's all the information that's presented in this one harmless looking slide. So before we move on, any questions about the material presented on this slide, this big overview slide? All right, if not, we can move on. Now, the membrane itself, there are other proteins than those that just go through it. So other than integral or peripheral or lipid link proteins, there's actually proteins that make up a skeleton, right? So the membrane also has a skeleton of protein around it. And the name of the protein for the skeleton is called spectrin, right? And the reason why they're called spectrin is because ghost, another word for ghost is a specter, right? So to discover spectrin, what scientists did is that they took our erythrocytes or red blood cells and they just basically popped them and let everything flow out. And what they saw this ghost shell floating around where it had the shape of a red blood cell, but there was nothing in it. And the only thing that was really left was this protein, which formed the shape of the cell. And so they named that spectrin. You can see here, it's a fibrous protein, just a bunch of alpha chains together. And the way they line up is that N to C terminus lines up with another N to C terminus. So that is one component of the skeleton of the membrane. There are other components as well. So in our membrane, we have these anion channels. These are proteins. And if you remember from general chemistry, anion is a negative ion. So that's a channel that lets through negative proteins or negative amino, not amino acids, ions, negative amino ions, sorry. Talking biochemistry, so I want to say amino acid really bad, but it is just ions. And attached to these anion channels, is this protein. So this is another protein called ankyrin. And ankyrin has kind of like this bent look, even though it's drawn as a sphere here. And what spectrin will do, and spectrin on this image are the red and blue. So this is spectrin, right? Alpha and beta chains as shown here. And they're kind of wrapped around like DNA. The way that spectrin is held in place in the membrane is they attach to ankyrin, which is attached to this anion channel. That's why ankyrin has its name. Ankyrin is named after an anchor. There's my very crappy anchor drawing. All right, I do not know how to draw. So named after an anchor because it anchors the spectrin to the membrane, which is all connected to this anion channel. Other proteins that are important in the skeleton are friend actin. So if you remember actin, that's your thin filament in muscles. However, actin has a lot of different utilities. So here, actin's not doing anything with muscle contraction. It's also there for stability. Topomyosin interacting with the actin. Again, we talked about that in muscle contractions. But what is interesting with spectrin and ankyrin is that you get different zones of your membrane. It's almost like create different fences. Right. So let me explain what this image is showing on the right. So on the previous slide, we talked about how, when we look at proteins in the membrane, they are mostly mobile. However, when you actually look at the movement of individual proteins, you'll see three different general types of behavior. One, the protein doesn't move. So it could be, so I said most protein moves, but some actually don't. B, there's some movement, but it's confined to an area. Or C, free movement. And the way to really explain why we have this type of behavior is because of the membrane skeleton. So the membrane skeleton, you can think of it as like kind of like a dispersed network of these proteins. And sometimes they form little gates and sometimes they have openings. So some proteins, like an A here, are actually attached to the membrane, to the skeleton of the membrane rather. And once they're attached, they can't move anymore, which tells us that that protein is critical to be there for the cell to survive. Otherwise, why would you take the time and energy to make sure it doesn't move anywhere? While in B, it could be, well, you just need to be in the general area, but you don't need to be in one specific spot. While in a protein like C, the idea there is you can be anywhere and do what you need to do. What's interesting about this model for the membrane, though, is when you look at a protein, let's take B, for example. When you watch the movement of protein B, and let's say, like, here's the cell, and you're watching protein B, you'll see it like just stay in one area, and then like move, and then like stay in one area, and then move, stay in one area. So although here it's shown that it has confined movement, it is possible that these proteins can like jump out of here into a new confined area. So we also often see that with proteins that are stuck in one area as well. But yeah, that's the different types of protein movements for proteins that are part of the membrane. And that's the actual proteins, the skeleton, that make up the membrane spectrum and anchoring. So any questions about the information presented on this slide? All right. So here's a good way to just test yourself if you understand what I was talking about. So multiple choice question, something you might see like on test 4. Which experiment listed below did not verify the fluid mosaic model of membranes? A, B, C, D, or E? Take a minute, review back to what you've written down, see if you can get this. Otherwise, I'll be back in a minute to go over this. All right, so hopefully you're able to at least make an educated guess about which one explains the fluid mosaic model. So remember, fluid mosaic model is what our topic is today. The membrane is movable. The things in it can move, and it's just made out of a lot of different parts. So let's see. Observation of lateral movement of membrane across the cell while anchored to a microscope slide. So we didn't talk about this, like I didn't say those exact words, but that did verify the models. That's kind of like what I was talking about when we did the FRAP experiment, where if you just hold it steady and you'll see things move. So yeah, that's what's happening there, where you just make sure the cell itself can't move. And if you just watch the membrane, you'll see the membrane move. So that's okay. And that's D as well, where that's FRAP. C, membrane fusion between mouse and human cells. Yeah, we did that experiment where we took those two cells, fused them, and then watched the proteins. You didn't do B though. So the mouse and human experiment is you fuse the cells. We didn't actually just put human proteins in the mouse cell membrane. I guess an argument against doing that type of experiment is, since you're putting a foreign protein inside of a cell, well, maybe it's moving because it's not supposed to be there, right? So that could be a criticism of that experiment. So that's probably why people didn't do it, but that would be B is your correct answer. It did not verify the fluid mosaic model. To speculate on functional reasons why some membrane proteins are anchored rather than free to diffuse laterally. So I answered this during my explanation, and this is a very good question, again, to come back to when you're studying for test 4 to see if you remembered what we went over in class. But again, if you're anchored, that means you're critical to be in that one location. If you're free to move, it doesn't matter where you are in the membrane to do your job. But if you're anchored somewhere, it's critical to be there for whatever reason. Maybe you need to interact with other proteins that are also there, or maybe you have to interact with certain lipids that are right there. But yeah, that would be the functional reason. Now, let's look at the actual lipids that make up the membrane. And we talked a little bit about this on Friday. But when you look at the lipids that make up the phospholipid bilayer, they are not the same on both sides. So let's look at this image to begin with on the bottom left. If we're looking at just total phospholipids, 50% of the lipids are found on the inner leaflet, that is pointing towards the cytoplasm, and 50% of the lipids are on the outside. That's good. So it's a 50-50 share. However, when looking at the proportion of total lipids, here we have that sphingomyelin, they make up about 20% of the outer leaflet, while 5% of the inner. Phosphatidylcholine, let's say like 22%, let's say like 8%. Phosphatidylethylamine, 25% on the inside, maybe like 5% on the outside. Phosphatidylserine, let's say like 9%, 0%. So I don't know if you remember from Friday, but I had a question of phosphatidylserine. I said, why is phosphatidylserine such a good marker for cell death when it's found on the outside of the cell? And I went over this, but here's an actual image that shows it. Now, phosphatidylserine is only on the inner leaflet when things are working normal. When things are not working normal, then that's a signal for something went wrong. So it's either a signal for death, or it can also start the chain reaction to start blood clotting, because if you have a wound, that means you've broken some cells open, and then you'll have phosphatidylserine just floating around in the bloodstream and in the local area. And clotting agents can recognize this free-floating phosphatidylserine, and they'll start recruiting other molecules once they bind to that to close the wound. So that's one reason why, the lipid leaflet is not the same on each side. So the question is, how does this happen? How do we have two sides be that different? If you remember that movement from one side of the lipid to the other side is very slow. And the answer is we have certain enzymes that move lipids from one side to the other, called flippase. You also have floppase and scramblase. So flippase, floppase, scramblase. And they each do something different. Flippase will move certain phospholipids. Floppase moves other phospholipids. Scramblase moves like 2 lipids at a time. You don't need to know the exact differences from them. You just need to know, what are their general purposes? And their general purpose is to move lipids from one side of the membrane to the other. You also have phospholipid translocases. Now these use ATP. So this is when you are moving against concentration gradient. So for example, sphingomyelin, if you want to move that from the inner to the outer, you're going against diffusion, natural diffusion, because normally you would want to be on the inner because this has a high concentration. So you would need to spend ATP to move that to the other side. Now, to make these lipids, the enzymes are integral membrane proteins of the ER. So the ER, if you remember your basic biology, also has membranes. So these enzymes are embedded in the membranes of the ER. Now, prokaryotes, they don't have organelles. So their integral membrane proteins are in the plasma membrane rather than the ER, and they just make their lipids right there. Us, we make them in the ER and then transport those out to the membrane bilayer for incorporation. Right. So any questions about the information presented here about lipids being more prevalent on one side of the leaf versus the other? All right. So here's a good review question that we talked about at the very beginning of class here. So a good way to just refresh yourself. FRAP, what is FRAP useful for? A, B, C, or D? So take a minute, review your notes on FRAP, and just ask for each of these actually. I guess this is a yes or no. So would it be useful for A, yes or no, B, yes or no, C, yes or no, D, yes or no? So take a minute, think about that, and I'll be back. All right, so hopefully you had some time to refresh yourself on FRAP. If you're still confused about FRAP, the 30-second reminder is put a fluorophore on proteins so you can see them. Hit it with a laser. Eraser. Part of the fluorescence goes away because you killed it. And then if proteins are mobile, this will slowly recover as proteins will just naturally diffuse in the area. So that's FRAP. So can you use it to measure the concentration of a protein in a cell? No, because it'll just, you could use fluorescence to see, how prevalent one thing is to another, but shooting it with a laser and then watching it diffuse, that's not going to tell you anything about concentration. So the technique itself, no, you can't use that. Could you use it for the lateral diffusion of a protein in a cell? Sure. You can see how fast proteins move in the cell. That's exactly what FRAP, one of the main uses of FRAP is. Could you use it to determine the speed of flip base? I guess I should have said the mechanism speed. And no. So flip base, all flip base is doing is it's moving a lipid from one side to the other. When we're looking at a cell, right, we're looking down on it. And let's say you fluoresce this cell, right? It has fluorescence. And then you move it to the other side of the membrane. It's going to be in the same location, just on the other side. So if we were looking at this fluorescence in a cell, it would be like, well, the fluorescence is right here. Flip base works. Okay, does not look like it moved at all. Because we can't, with like a microscope, that's going to be very hard to see something on that level of detail. And especially with fluorescence, you can't tell, like, is it on the top leaflet or on the bottom leaflet for a lipid? So you can't do that. D, could you do that for the lateral diffusion of a lipid in a cell? Yeah, probably. You just have to put fluorescence on your lipids. That might be a problem, but should be theoretically possible to do that. So any questions about FRAP or what it could and could not be used for? All right, if not, we can move on. Now, although we've been saying that the membrane's a mosaic and it's just made out of different parts and that it is fluid, there are parts that are kind of like stable and they're organized. And they're called lipid rafts. Excuse me. When you look at the plasma membrane, and this is supposed to be cells, you'll see that there are areas of organization where you have some high concentration of specific lipids or specific proteins. And this is what we're calling lipid rafts, right? It's just like an island floating in the lipid sea, an island of stability in the sea of lipids. And when we look at these lipid rafts, they're very ordered and almost like a crystal. So if you look at a salt crystal, for example, it is just a very ordered repeat of like NaCl, NaCl. Or a protein crystal is just repeating that protein in every single, the same way over and over and over again. So lipid raft, what that's saying is that like it's organized and it's repeating. When we look at lipid rafts, there are certain proteins and molecules that are generally aggregated together. So our GPI linked proteins If you remember, GPI-linked protein is the protein with the big ganglioside on it, the big sugar. And these proteins are generally attached to our lipid rafts. Maybe they're used for intracellular signaling. That's the best estimation of what they're used for. But it's also how different viruses, like the flu, measles, Ebola, HIV, that's how they get into our cells, is that they'll attach themselves to this island of stability, this lipid raft, and that's how they'll invade the cell, through these lipid rafts. So they're important, but they also leave us vulnerable to attack from viruses. So one more review question, and I'm trying to, this PowerPoint has been different than the other ones in that I'm staggering the review questions after we cover the material. But this goes back to a previous topic we talked about. So the biconcave shape of erythrocytes, or, erythrocytes, when you look at them, they look like a donut, kind of. That's really bad, but it's like a circle with like a dent in it, almost like a donut. And that shape is there so it can diffuse oxygen very fast. How does that happen? So take a second and review yourself about how the membrane of a red blood cell might get its shape based on what we talked about. All right, so hopefully you had a second to remind yourself, and that is going to be C. So spectrin is what makes up the cell membrane, and it's also what's going to give our red blood cells their shape. And just to go back to one thing, I was just looking this up, something I probably should have looked up earlier, but let's add to this list. Influenza, measles, Ebola, HIV, COVID-19, all localized lipid rafts to get into your cells. So they all attach here, and then they use these little lipid rafts, and then they spear your membrane with like a needle and then inject their RNA inside the cell, and that's how they replicate. So any questions so far about anything we have covered? All right, so it's about 250. So let's take a look here. Yeah, okay. So I'm going to leave this question as a question for you to review on your own, so you can get it. But on Wednesday, we're going to cover this process, the secretory pathway. This will take me probably 15 to 20 minutes to go over what it says, so I don't want to start that here. And then we have a bunch of questions that cover the secretory pathway. So we'll leave it here for right now. Reminder, test due next week, Monday. I'll put a homework up that goes over our the material that we covered. Since this homework that I'm going to assign is not part of test 3, I'm going to give you an extra 2 days to do it. So The homework I'll assign for this PowerPoint, that'll be due in 9 days. And I'll make that clear on the dates of the Wiley assignments, where I'll say like due March or April 14th, part 1, part 2. So yeah, there will be a homework assigned, but it's not due on Monday. It'll be due next week, Wednesday. Otherwise, I think this is a good place to stop. As always, email me if you have questions. Otherwise, hope you learned something today, and I will hopefully see you on Wednesday. Take care.